All right, welcome everybody to our final tutoring session for the anatomy. We're wrapping up all of the remaining content that we have to cover prior to the local competition reviews. The local competition review is on January 11th. That's an interactive review. We're working on something fun for you guys, so definitely come back for that. I do have a couple other announcements. One, registration closes on January 12th. So if you have any friends that still might want to register for the Anato-Bee, they do still have some time. That being said, we're waiting until registration closes in order to assign you to local hosts for the local competition assignments. An email about that should be coming out soon just to kind of get everybody um, on the same page. I know some people are a little bit worried about how that's going to work. So don't worry. We know that you're registered. We got you. You will take the local exam. So we're good there. We did have a student create a review for you guys. And so I'm going to go ahead and put that in the chat, and then we're going to go ahead and post it in our resources as well. So it's all on skeletal anatomy. And part of this is to help you kind of get used to what the Google form interface looks like so you can kind of practice and feel a little bit more prepared for when you take the local level of the exam on Google Forms. Another thing to let you all know about is our Anato-Bee research survey. So I'm going to go ahead and put that in the chat as well. So this is part of the Anato-Bee. We're looking at motivations and persistence in STEM education. You do not have to take the survey to be part of the Anato-Bee. It's optional, but just kind of putting it out there. So those two links are in the chat. And otherwise, I think that was all of our announcements today. Dr. Frank, did you remember anything that I didn't say from our
No,
pre-discussion?
I
No.
th- think you covered everything.
Awesome. All right, I will go ahead and pass it over to you, Dr. Frank, to moderate our session.
Okay, great. Well, welcome back everyone for week 9 of our tutoring sessions, and I will serve as your host one more time for the evening. And we have again 3 very talented medical students here who are going to serve as the tutors for this evening. And our first subject is going to be anatomy of the immune and endocrine systems. And we have with us this evening Deepthi Sathanarayana. I hope I got that close. Okay, so Deepthi is from Sammamish, Washington, and graduated from the University of Washington with a bachelor's in biology and a minor in global health. In her gap years, she spent time as a medical assistant in an abortion clinic and Seattle Children's Oncology Department. She is currently in her second year at the Pacific Northwest University College of Osteopathic Medicine. Her primary interest is in pediatrics and OB-GYN. She enjoys singing, baking, and painting. And with that, I give it to you, Deepthi, to go over the anatomy of the immune and endocrine systems.
It's like you're still muted. And if you could make it full screen for us, that would be helpful as well.
Mhm. Is that better? Okay. Thank you, Dr. Frank. So these are the lecture objectives. So by the end of this, you should be able to describe the organization and function of lymphatic structures, including nodes, vessels, ducts, and capillaries, and identify the major groups, including cervical, pectoral, axillary, and inguinal lymph nodes. And for the endocrine system, be able to list the major endocrine glands and describe where they're located as well as their general functions. So the first question is just the blank system is a network that helps return extra fluid from, and I cannot, oh, if we can have the answers in the chat, labeled 1, 2, and 3, that would be good. I'll just move on to the answers. So the first one should be pretty simple. Simple. The lymphatic system is a network that helps return extra fluid from tissues back into the bloodstream, and it plays a major role in the immune defense, and it follows a general pattern, and we're going to be looking at that pattern next. These are just some pictures to help you figure out. Okay, it goes from the capillaries to the nodes, then to the trunk, then to the duct. Okay, so a practice question. Which of the following best explains why lymphatic vessels contain one-way valves? A, to increase blood pressure within the lymphatic system. B, to allow lymph to move using cardiac contractions. C, to prevent the backflow of lymph in the absence of a central pump. And D, to filter pathogens from the lymph.
I'll go ahead and end the poll.
Okay. So the right answer is C, to prevent the backflow of lymph in the absence of a central pump. So, unlike the cardiovascular system, the lymph doesn't have a central pump like the heart. The lymph just moves in one direction, following a general flow pattern from the tissues back to the venous circulation. And it has three major jobs to maintain fluid balance, support immune surveillance, and transport absorbed fats from the GI tract. And that's through the chyla, cisterna chyli. Yeah. Okay, so another question. So going from lymphatic capillaries, what's the next space that the lymph travels through? We can answer in the chat. Oh, I gave that away. Whoops. So it goes from capillaries that you see right here to the lymphatic vessels. Where does it travel to next? Yeah, lymph nodes. Someone said it in the chat. That's correct. And as you can see, there's like multiple lymph nodes. We are taught in the physical exam to check the lymph nodes here. So pre-auricular, post-auricular, occipital at the back, submental, and the tonsillar lymph nodes, as well as supraclavicular. And that one's supposed to hurt a lot when you're checking for those because you have to go really deep inside. Okay. Next, we go to the ducts. Yes. And then after that, where do we go back? Where does the lymph flow? From the ducts to the venous system. Yep. Good job. All right. Another question. Which structure is responsible for collecting excess interstitial fluid and begin its transport as lymph?
Close the poll in 3, 2, 1.
Okay, most of you got this one correct. It starts at the lymphatic capillaries. And so the lymphatic capillaries are just tiny blind-ended tubes. So it's just one-way flow, and they have thin walls made of single cells, single layer of endothelial cells, and they loosely overlap, allowing fluid to, flow through easily. And it has um, pathogens, sometimes cellular debris, as well as large proteins that can't be absorbed by the capillaries. Um, Next slide. The lymphatic vessels are larger channels that the capillaries just, merge into. And again, they're all one way to prevent backflow. And these vessels move with skeletal muscle contraction and changes during breathing. And their main function is to carry lymph from the tissues to the body center and then transport the lymph through the nodes along the way. And then for the nodes, again, there's several. The cervical lymph nodes, axillary lymph nodes, where it's by the armpit, inguinal ones, and these are all like bean-shaped organs, and they can contain immune cells, including B cells, T cells, and macrophages. And they're found in places where pathogens commonly enter the body. And again, it's to filter lymph and trap pathogens, and they will activate immune responses when needed. And that's why when your physician, feels around your neck, they're looking for those swollen lymph nodes. Next question, which lymphatic duct drains lymph from the left arm, left side of the head and neck, and both lower extremities?
End the poll in 3, 2, 1.
Okay, so the right answer is B, the thoracic duct. The thoracic duct drains lymph from most of the body, basically everything except for the right upper quadrant. As you can see in this picture here, it begins at the cisterna in the abdomen and empties into the left subclavian vein. The right lymphatic duct drains the right head, neck, right arm, and upper chest, and empties into the right subclavian vein. And together, these ducts return filtered lymph to the venous circulation. And yeah, that's about it for this slide. The next one should be a question. Um, okay, so it's endocrine glands secrete hormones blank into the blank to act on distant targets, which are ductless, where exocrine glands secrete substances like sweat, saliva, milk, or digestive enzymes through blank onto body surfaces or into cavities. If we can have a 1, 2, 3 in the chat, that would be good. So endocrine glands secrete hormones directly into the bloodstream and act on distant targets, and they're considered ductless. And exocrine glands secrete substances through ducts onto body cavities or surfaces. Okay, so these are the major endocrine glands: the pineal gland, pituitary gland, thyroid, parathyroid, adrenal, pancreas, and ovaries/testes. And we're going to be going more into detail. Yeah, just focus on the location and primary function when I go through the next few. So the pituitary gland is considered the master gland or the central gland because it regulates growth, metabolism, reproduction, stress response, and water balance. And there's two parts to it, the anterior pituitary and the posterior one. And a mnemonic for the anterior pituitary gland and what it secretes would be flat peg. So follicle stimulating hormone, luteinizing hormone, adrenocorticotropic hormone, thyroid stimulating hormone, prolactin, endorphins, and growth hormone, whereas the posterior pituitary secretes only oxytocin and ADH. And also, the posterior pituitary only stores and releases the hormones. It doesn't like synthesize these hormones. That's about it. And the thyroid is at the C5 level anterior to the trachea, and it synthesizes and releases the thyroid hormones. So that's about it. They regulate the basal metabolic rate. It's critical for energy use, heat production, and growth. The adrenal glands are on top of the kidneys and they produce hormones like cortisol, aldosterone, and adrenaline. They all regulate metabolism, blood pressure, stress, and water balance. So the pancreas is a retroperitoneal organ, which means it's fixed to the back of the abdomen and doesn't move freely like the intraperitoneal organs. And it's posterior and inferior to the stomach. It behaves as an exocrine gland, but also as an endocrine gland. The exocrine one is just it produces and releases lipase, protease, and amylase, which are essential for breaking down food. And then the endocrine one produces hormones like insulin, glucagon, and those two regulate blood sugar levels. Next one, ovaries and testes. The ovaries are in the pelvic cavity, lateral to the uterus, and produce and secrete estrogen, progesterone, and inhibin. And testes are within the scrotum and produce and secrete testosterone and inhibin. So, which endocrine gland regulates many other endocrine glands?
Close in 3, 2, 1.
Okay. So, most of you got this right. It is the pituitary gland. It's in charge of metabolism, growth, and so much more. So, it does influence a lot of the other glands. I don't think I have any more questions. These are my references. Um, yep. Thank you.
Do we have any questions for Deepti? Do we need to know any of the specific hormones?
Um, I think according to the learning objectives, you don't need to know the specific hormones, but I could be wrong.
Dr. Mapes, did you want to?
Yeah, so for now, you want to just know the names of the glands and where they're located in the body. Yeah, and I mean general function is good as well. So knowing that the thyroid gland regulates metabolism or something like that is good.
Also, endocrine versus exocrine function. So,
Absolutely.
any other questions? We have a little time, so if you have anything, this is the last session. All right. Well, um, I guess that
Nice.
will be it then. Oh, we do have a hand. Uh, Go ahead.
Hi, sorry, this is a random question. Can you tell us how to pronounce your last name? Sorry, I'm so curious.
Sure. It's Satyanarayana.
Okay, thank you. Sorry, that's just so cool.
Thank you. Yep. Jacob?
Hi. When you were talking about the lymph nodes, so when the doctor checks if they're swollen, would that mean that you're like fighting an infection or something, like your white blood cell count's higher?
I think so, but sometimes they could just be swollen and you wouldn't have an infection. Um, I think it's just checking the size of them too, not just like if it's swollen, but um, yeah. Dr. Rapes, do you have?
Okay, thank you.
Yeah, I would say, if you go in with a sore throat, usually they're going to check to see if your lymph nodes are swollen when you go to the doctor. But at that point, you're probably also going to get strep culture and a bunch of other things. So it's not really necessarily diagnostic, but sometimes people will notice pain in their lymph nodes. So someone might have like armpit pain that's really bothering them. And that could be an indication that they might have breast cancer because the lymph node is getting some cells from the breast as the lymph is draining, and so they're actually getting a lump or that pain in their armpit just because of the way that the lymph is draining. So if you do have swollen or painful lymph nodes, it's definitely something to get checked out, but it's kind of one of those things that's in a series of multiple other symptoms that a doctor might use to come to a conclusion about your diagnosis.
Okay, got it. Thank you.
Mhm. Yeah, and then using something like OMT, deeply, you
Hmm.
could help drain lymph nodes, right? If you're having a lot of pain, uh, wherever that is.
Yeah. Um, yeah, for this one, the otitis media, we use the gall breath technique. So it's just bringing the pre-auricular lymph nodes back to, um, I guess, the heart or, yeah, the center of the
So
body.
an ear infection,
Yeah,
yes.
sorry,
Yeah.
ear infection. Yes.
In my world, we used to call that gland stripping, but basically you're fostering the flow of lymph from those vessels back towards the drainage back into the arterial or to the blood system, getting it out of the external back into the internal. Great questions.
There's another question in the chat. Do you want to?
Yes. Yeah, the other nearby lymph nodes would get, you know, swollen because it's all part of the same like drainage system. So if you get, if like one part of the system, I guess, misses the trapped pathogens, the other part would catch it. So it's not like um, only that only the ones closest would be the ones swollen. Um, but I think that's mostly the case. Ones that are closest to the pathogen would be swollen. Yep.
Cool. Great
All right.
questions.
Yes. Any other? Okay. Well, all right. Well, thank you, Deepthi, and great questions, everyone. Um, we will move on to our second section then. And so tonight we have Davina Prasad with us from Drexel University College of Medicine. Davina is going to go over histology of the lymphoid organs and integumentary system. So Davina was born and raised in Long Island, New York. She graduated from Brooklyn College in 2019 with a BS in biology, BA in chemistry. She then earned her MPH from UPenn in 2021 and MS from Drexel in 2023. She is now a second-year medical student at Drexel University. In her free time, she enjoys reading, trying new recipes, and spending time with friends, her mom and brother. Academically, she is interested in anatomy and embryology and wishes to specialize in OB-GYN or breast surgery. Davina?
Hi, Dr. Frank. Happy New Year. Hi, Janelle. Happy New Year, everyone. So excited to be here. Let me just share my presentation. Let's see. Okay. Dr. Frank, are we good? Can you see? Okay, great. Let me try to go. Is it full screen? I think so. Okay, great. Hi,
I think
everyone.
you're good.
Okay. Well, hi everyone. So as Dr. Frank said, I'm going to be doing the histology section. We're going to be talking about the integumentary system and lymphoid organs. So we'll start if this would go. So just our learning objectives. So talking about two major layers of skin and their histology, location, function, and then the histology of the hypodermis as well as its location, function, and yeah. Okay, so I will be doing mostly a question format. If you have any other questions or points of confusion along the way, you can drop it in the chat. But for now, we'll start with the first question here. Dr. Frank, would you like me to? Oh, thank you for running the poll.
Already got it.
Thank you.
I'm here to help.
All right, yeah, great. So we have about 26%. I think we could probably close when we're at about 80, hopefully. Okay. This is great. All right. I think for time's sake, just because I have about 10 questions to go through in the next few minutes, I will end it here. So most of you chose answer C, but we were a little bit scattered, so I will go over that. So okay. The correct answer was C for the hypodermis, but I will just quickly go over what A and B were. So A refers to the epidermis, which here we can see that the hair shafts, as well as the pore of the sweat gland ducts, are seen there. So this is the uppermost layer. Then we have the dermis, in which we have all these other structures that you can also see here. So the erector pili, hair follicles, the oil glands, as well as some of the sensory portions. And then lastly, here on the most bottom portion, we have the hypodermis. So I think the next slide is, oh, I had animations. I didn't realize that. Okay, for our next question. Which layer of skin is found in thick skin but not thin skin? This one is a little tricky, but we will go over all of the layers. Just give it a few more seconds. Okay. All right. I think we can close it there. All right, so we have an even split between choice A and B with some saying C and E. So let's just go over this. So the correct answer was choice B. So that would be the stratum lucidum. So just to review, so we have from the top to the bottom, we have the stratum corneum, stratum lucidum, stratum granulosum, the stratum spinosum, and then the stratum basale. I think I do have a memory aid somewhere on here. Maybe we'll get to it. But one way to remember this from top to bottom is I usually like to say, come, let's get sunburned. It's kind of just a way to remember. Dr. Frank taught me that. It has to do with the skin, and you kind of can remember the layers from top to bottom. So the stratum lucidum would be found in thick skin and not thin skin. And then I think the next slide, we're going to be talking about thick versus thin. So as we know, not all of our skin is the same. Thick skin, which is found on the palms of the hands and the soles of the feet, are more prone to wear and tear. Just it undergoes a lot more mechanical force, friction, and yeah, it needs to have a thicker layer of skin. So that was where you would find the thicker layer of the stratum corneum, as well as stratum lucidum. And then thin skin would be other areas of the body. So like the face, the arms, the neck. So really when we think about thick skin, we think about palms of the hand and soles of the feet. And then here we're just talking about a little bit of the histology and function. Oh, so here is our memory aid. Come, let's get sunburned. And like we said, the stratum lucidum is only present in thick skin. Okay, now which of the following layers contains lamellar granules? Great. I'm loving this. Okay, great. Oh, all right. Looks like we've kind of tapered off there, so we can end the poll. So most of you answered choice B. And that would be correct. Oh, sorry, choice C. I'm so sorry. Choice C. Choice C is correct. So this one I kind of just remember as in the granules are found in the granulosum. So sometimes the words are very kind and useful in remembering. So the stratum granulosum contains, as you can see here in this picture, the dots that you see here kind of around the nucleus and throughout the cell would be the lamellar granules. And then this is just a slide about the integumentary system. So we're looking at an H&E stain here of thick skin versus thin skin and how that might look in more pigmented skin versus lesser pigmented skin. So here we see in thick skin that this layer here of the stratum corneum is very, very thick. And that is in contrast to here, the thin skin. Also here in darker pigmented scales, we see that here this basal layer of cells has more pigment compared to the thin skin with light pigmentation. So this tends to have a more well demarcated kind of layer of cells that contains that melanin pigment. Okay, question 4. Which of the following types of skin shown in the images here would contain hair follicles? So it's a little tricky, but think about the images that we saw on the previous slide and what we talked about on the first slide. Great. Okay. And yes, A would be the left image and B would be the right image. Okay. All right, good. We got a lot of answers. Great. We can close this off. All right. So most of you chose answer B, which is correct. So here on the left, we have thick skin, and on the right, we have thin skin. So remember that thick skin would be found on the palms of the hand and soles of the feet, and those are not surfaces on which we typically grow hair. So that was one way to think about it, but also thinking back to that first image that I can go back to at the end. You see that in thin skin, that is where we would have the hair follicles, as well as the sebaceous glands, etc. Okay. All right. So this one, well, I was going to accept comments in the chat and then I realized that I can't see the chat. Oh, did I stop sharing? Are we good? I try to see the chat, but I cannot. Oh, there we go. All right. So it looks like I cannot see the chat in the same way, but sorry, Dr. Frank, I like lost my gallery. Can you guys still see my screen?
Yes, you're good.
Oh, okay. Well, I kind of gave it away, but if just in case you were distracted and didn't see this, um, what layer is marked in red and what layer is marked in green? If you could just put it in the chat, that would be great. All right. I see the answers populating in the chat, but I cannot actually see the chat. So I will just move forward for time's sake. So red would be the stratum corneum and green would be the epidermis. And we could see here that in thick skin, we have a much larger stratum corneum delineated here in red compared to thin skin, as we discussed 2 slides before. And then in green, we have our epidermis. Great. So question 6, identify the type of connective tissue shown here that is found within the reticular layer of the dermis. Okay. Oh my gosh. Okay. Hopefully no one saw that. Okay. Yeah, at 80%. All right. Great. So most of you chose answer D, which is dense irregular connective tissue, which is great. So this is a type of question that kind of ties in our earlier lecture on epithelium that we talked about all the way in September, and our topic today. So here, this red arrow is just pointing to the dense irregular connective tissue. And again, that's found in the reticular layer of the dermis. Okay. All right. And here we're just talking about the dermis, histology, and its function. So here you can see that where you would see these ridges, that's what we consider the papillary layer. And then at the bottom, the question that just asked to classify this tissue, the dense irregular connective tissue, would be the reticular layer. So this is the same thing shown over here, that this area kind of where you see these ridges would be the papillary layer, and then below that would be the reticular layer. Okay, just a quick slide on what is the dermis. So when we're talking about the dermis, it's the layer of tissue where you would find things such as the hair follicle, roots, glands, these sensory organs such as the Pacinian corpuscle that senses vibration and deep pressure. So that is found in the dermis. And then the hypodermis, it's notoriously difficult to kind of recognize, but generally it has more adipose tissue and is well vascularized. Okay. All right, so now we're just going to shift gears into the lymphoid system, and the learning objectives for that is just to compare the organization, location, function of diffuse lymphoid tissue versus lymphoid follicles versus lymphoid organs. Okay, so here we're talking about, we're going to be going over the histology of the thymus. So the thymus is found under this layer of fat tissue here. Involution of the thymus tends to happen around puberty, but we're going to be talking more about structure and function. All right, so question number 7, which of the following labeled areas indicates the cortex of the thymus? Okay. I'll just wait till we get to 80%. Okay. I have about 3 more questions, so I guess we can end the poll here just to stay on schedule. So there was a pretty even split between A and B, but we'll go over it. So the correct answer was A, which would be the cortex. So the cortex is the outermost layer that you would see here. So it is slightly more pigmented than the medulla, which you'll see deeper here, which is lighter in color. I won't give away too much though, because I think we have another question on that. Oh, okay, here. So just talking briefly about the function of the thymus. So it is the site of T cell maturation, and T lymphocytes, as you know, are really important in fighting off infections and the overall function of the immune system. Moving on. So question number 8, which of the following best describes the primary function of lymph nodes? Can take a few seconds to just read through the answer choices. Mm, 39. I'd love to get a couple more answers and then we can go over this one. Okay, we got 100%. Or 75%, which is good enough for me. So we'll end the poll here. So we have a majority of you chose answer C, with some choosing answer A. So the correct answer was answer choice C. So the primary function of the lymph nodes is filtration of lymph and initiation of those adaptive immune responses. I just want to clarify. So answer choice A, which was talking about filtration of blood and removal of aged red blood cells, was a little bit of a trick, but that would be the function of the spleen. So that is one thing to kind of just be mindful of, that the spleen filters blood while the lymph nodes will filter the lymph and also play that role in adaptive immunity. Okay. All right. So just talking about the lymph nodes again. So we have this outermost layer here, the cortex, and then we also have within this connective tissue capsule here, we have the subcapsular sinus. So underneath the capsule, we have the subcapsular sinus. So the language kind of helps here. We also have the germinal centers here that you can see with these arrows here. So that would be the site of the T cell maturation. Okay, and then question 9. So we have identify the feature of the lymph node that's shown within the yellow circle. Again, I said put answers in the chat. I cannot see the chat. So we'll just give it a few seconds and then I will go over the answer.
We also have a question from the last slide as well.
Okay. Let me see. A would be spleen's function, right? Yes, that is correct. Okay, so for question 9, just for time's sake, I'm just gonna keep going. Okay, so the yellow dotted circle was indicating the germinal center. So you can see here that it kind of forms this concentric, there's this layer of more pigmented cells, and inside that we have this lighter area, so that would be the germinal center. And then just kind of recapping that this outermost layer here is the cortex, and then with below that we have the medulla. And this is just the same thing, the germinal center zoomed in so that you could see that a bit clearer. And then briefly just talking about histology and function of the spleen. So as you all mentioned, that the function of the spleen would be filtration of the blood as well as destruction of red blood cells when that is necessary. The spleen here is found underneath the diaphragm. I'm sure that is more of an anatomy thing. But yeah, here we have some histology about the spleen, and I will go forward because I think we have a question. Great. So which of the following best describes the primary function of the spleen? Which we've kind of talked about, but okay. Great. I love this. I love when the other questions help us with the next ones. All right. Okay, so I think we can end it there. Um, all right. So most of you chose answer C, which is correct. So again, the spleen is going to be doing filtration of the blood. Remember that the lymph nodes would be the function, the organ correlated with the function listed in answer A. Production of erythrocytes in adult life would be the bone marrow. That's the only other one I think we wanted to talk about here in this lecture. Okay, so just talking about lymphoid nodules and diffuse tissue. So the lymph nodes are the kind of discrete organs that we can actually palpate, we can touch, but also throughout our body, we have other areas of immune tissue known as diffuse tissue. Specifically, there's the gut-associated lymphoid tissue known as GALT. So here, I think we just talk about the location of the tonsils. So a lot of people know have either had their tonsils removed or have had some lifetime experience with the tonsils. So these are referred to as the palatine tonsils, which get swollen during the presence of an infection. And then here, just briefly, the histology of the palatine tonsils. So we have these crypts here, which are kind of these breaks in the tissue. And below that, you would see stratified squamous epithelium. Palatine tonsils also have germinal centers, similar to the lymph nodes and other immune organs. And here we have pyre patches, which are pretty special. Okay. And then these are my references. I think those were all the questions that I had for you all. Okay. Does anyone have any questions for me? I'm just going to check the chat. I'm so sorry I couldn't see that before. You guys did really great. Thank you for participating, even though I couldn't see the chat. Okay. 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 All right. I guess we're so good. Well, thank you, Dr. Frank, for
Yeah,
running the
yeah,
polls.
yeah.
Thank you, everyone, for participating because I know, like myself, we're all coming off of the holiday schlump. um, yeah, I'll stick around for a couple minutes if anyone has any questions. I'll also put my email in the chat if anyone has any questions, but I think that's it.
Okay.
Thank you.
There no more questions for Davina. Thank you very much, and
Thanks, Dr. Frank.
appreciate all your help. Love the mnemonics. Okay. Well, I don't see any more questions, so we'll move on to our last section, and this will be embryology of the head and neck. So with us tonight is Savannah Gregory from the Edward Via College of Osteopathic Medicine, who's going to discuss this with us. So a little background on Savannah. Savannah is from Nashville, Tennessee, and graduated from the University of Alabama at Birmingham, majoring in biology with a minor in Spanish. She is currently a second-year student in Auburn, Alabama, with an interest in anesthesiology. In her free time, she enjoys reading and spending time with her cats and budgies. Her favorite part of anatomy is learning the vasculature and how the body is one connected unit. So connecting to the other two sessions, Savannah, let's connect this and bring it on home.
Hey everybody, thank you so much for having me. I'm gonna go ahead and try to share my screen real quick because I am sometimes technologically challenged. Okay. Is that showing up for y'all? Yes. Okay, perfect. Is
Can you move your cursor to see if we can see it? Yes, good. Okay.
Okay, is it still there now? Okay. All right, everybody. Well, thank y'all for having me on tonight. I'm excited to talk to you about a little bit of embryology. It's just like we said before, I know we're all coming off the holidays, but I hope everybody had a great holiday season and is excited to start this new year. So with that, let's go ahead and kick gears into embryology of the head and neck. So before we get into the learning objectives, I'm actually going to start this off with a quick question. And so we can open the poll. And y'all just feel free. It says, which of the following embryonic structures is most responsible for developing into the adult head and neck? Okay, I'm going to go ahead and end this poll. Great. So the correct answer here is B, which most of y'all kind of had more of an even split. So the pharyngeal apparatus is actually what's going to develop into the adult head and neck structures. It's sometimes also called the branchial with an L. So the branchial apparatus or the pharyngeal apparatus, they're the same thing, two names for the same thing. So the neural tube is what's going to eventually become like your central nervous system, so things like your spine. We kind of talked about that in an earlier session. So for head and neck, it will be the pharyngeal apparatus. So with that, let's talk about the lecture objectives. Tonight, we're going to be talking about what exactly is a pharyngeal arch, what is a pharyngeal pouch, and what is a pharyngeal groove. So with that, we can go ahead and one of the hardest parts about embryology, which I'm sure y'all know by now, is kind of visualizing things in space. So let's take a moment and go ahead and do that. So what we're talking about here with head and neck development is actually occurring within the first 8 weeks. So there's a lot of things that go on at the same time. And we're just kind of taking like a snapshot of specifically the head and neck right now. So embryogenesis is referred to as the development of the embryo from weeks 1 to 8. And the head and neck structure is going to be this whole thing. If you can see my mouse, what I'm circling here. This is the pharyngeal apparatus. Right here, we're looking at an embryo kind of laterally. So this would be like on the side of the embryo. And this image that we're seeing here is almost like a top-down view. It's almost like we took a slice and we took this slice right here and then we laid it flat on the table. So we're kind of looking at this slice here flat on a table. So some of the things you're going to see here are 5 pharyngeal arches, which you can also see on the lateral side here, 4 pharyngeal pouches, which are going to be on the inside, and then 4 pharyngeal grooves. And as we continue through the PowerPoint, that'll become a little more clear. Of course, there's a few more things in here. So each of these arches has some vasculature, nerves, arteries, but this is just how we're going to be visualizing these things in space. I don't know if I can see the chat, so if there are any questions, don't hesitate to ask. So let's go ahead and start question 2. During which week of embryological development do the head and neck structures begin to form? So we kind of talked about this a little bit, but all right, I'm going to go ahead and end the poll. Yeah, you guys were right. It is B. So it's going to be during week 4. So again, we talked a little bit about, let's see here. There it goes. We talked a little bit about that. Embryogenesis is those first 8 weeks, and specifically the head and neck will begin developing around week 4. This is when your pharyngeal arches are going to first start appearing, and you can see them here. They're labeled by Roman numeral, but you've got 1, 2, 3, 4, and 6. So initially you do have a pharyngeal arch 5, but it kind of just goes away on its own. It doesn't really develop into anything. So pharyngeal arch 5 isn't really considered when we're looking at the overall structure of the embryo when in embryogenesis. So we actually label it 4 and then 6 because arch 5 is going to go away. Around weeks 4 and 5, that's when your pouches and your grooves are going to appear. The grooves can also be called clefts. They're going to be what runs in between your pharyngeal arches. You can see them here, 1, 2, 3, and 4. The pouches are on the inside, so you can't really see it from this view because this is more of an external lateral view. But then once you get to weeks 6 and 8, that's when your structures are going to start differentiating. This is going to be the cranial portion, so the primitive head region. And then this right here is actually that neural tube that we talked about earlier that's going to eventually start differentiating into your central nervous system. Okay, question 3. Which embryonic tissue layer helps to form the core of the pharyngeal arches? Okay, I'm gonna go ahead and end this poll. Okay, so we're looking at like we're pretty divided, and that's awesome because that means this is an opportunity. So the embryonic layer that's going to form the core of the pharyngeal arch is actually going to be the mesoderm. Now, when it comes to the pharyngeal arch, I kind of call him the jack of all trades. So the pharyngeal arch is going to be this portion. So remember, this is kind of this red line right here is showing you where we cut. If you remember earlier, I said it's like we're looking at it lying flat on a table. So now we're looking at the arches, the clefts, and the pouch. So this is going to be the external, the outside, and this is going to be the inside. The arches are made up of mesoderm internally. So this little gray area on the inside, this is all going to be mesoderm, as well as some neural crest cells, mesenchyme. And the outside here is going to actually be ectoderm. So when I said what makes up the core, that core is going to be this mesoderm. Now, when the arches are developing, they do develop cephalad to caudad, which just means they develop from the head down to the tail. And when we talked about this earlier, the reason why it goes from 4 to 6 is because pharyngeal arch 5 actually does disappear. There are no adult structures that come from it. Internally, we talked about the mesoderm, but let's just take a little quick minute to talk about the mesenchymal neural crest cells. Now, mesenchyme was a word that I didn't truly understand for a while. It was a little confusing to me. So just to break it down, when somebody says mesenchyme, what they're talking about is just either a neural crest cell or a mesoderm migrating to an area. Mesenchyme can be just one layer or it can be multiple. So for example, this could all be just mesoderm, or it could also be mesoderm and neural crest cells. It's just talking about a migrating tissue component. And that's what it's saying here. It just describes loosely organized connective tissue. I don't think you have to get really bogged down by this definition. I just think it's an important terminology just to have some exposure to here. I don't even really think it's in your learning objectives, but I just wanted to point it out just in case you did any reading on your own and you saw that terminology. Okay, question 4. Which embryonic tissue layer helps to form the pharyngeal pouches? Oh, I just saw a question. I'll answer your question after we finish this poll. Okay, I'm going to go ahead and end the poll. All right. So it does seem like most of you picked D, And that is the correct answer. So the pharyngeal pouches are made of endoderm. It makes sense. They are on the inside. So a mnemonic that I like to use is that, and this is completely silly, I don't know why, but whenever I think of a pharyngeal pouch, I also think of a baby kangaroo in its mother's pouch. And they go endopouch, and that is how I remember that pharyngeal pouches are made of endoderm. So whatever sticks, figure out your tips and tricks. For me, that's it. And now when you think of a pharyngeal pouch, I hope you'll also think of a kangaroo. Yes, so these are formed on the inside of the pharyngeal apparatus. We have our arches, our grooves, and then our pouches. There are only 4 pouches, and the first one lies in between pharyngeal arch 1 and 2. So you can see it color-coded here. They're going to kind of be aligned with the clefts. So since we have 5 total arches, we're going to have 5 clefts and 5 pouches. Let me go ahead and answer this. Oh, yes, perfect. The question was answered. I'll just say it out loud. So, yes, arch 5, it doesn't necessarily atrophy, it does go away just as things are developing. Certain things are going to proliferate or get bigger. Certain things are going to get smaller. Certain things are going to kind of mesh together. And just in all of that process, it's just something that naturally happens, that arch going away. All right. Question 5. Of the 4 pharyngeal grooves, 3 of them are going to disappear, but 1 groove remains, and it's going to give rise to the epithelium of the external ear. Which of these grooves is the one that remains? I think it also gives rise to the external auditory meatus, but that's okay. Okay, I'm going to give it just a couple more seconds here. All right. I don't think this is explicitly in the lecture learning objectives, but the correct answer is A. It's going to be groove 1. We're just going to talk about it here so that we can kind of talk a little bit about the cleft development. So these clefts, otherwise known as grooves, there's going to be 4 of them here. All of them are going to disappear except for this first one. The reason why is because this arch right here, this is your second pharyngeal arch, as it grows and develops into its adult structures, it's going to actually, just like we talked about how pharyngeal arch 5 disappears, pharyngeal arch 2 gets bigger, and it's actually going to combine and in a lot of intricate processes is going to kind of overtake this area, and it's going to fuse together so that these grooves kind of go away. They kind of regress as what was stated earlier. But this groove is above pharyngeal arch 2, so it stays and it develops into portions of our external ear. The grooves are made of ectoderm, so that's all of the external. That's a great way to remember it. It's external, it's outside. We're looking at this lateral portion of the pharyngeal apparatus. It's all made of ectoderm. So even though these are the arches, remember, the inside of the arch, this gray area here, the core is made of the mesoderm, but all of this lateral portion, so the grooves and then the external pharyngeal arches are all made of ectoderm. Hopefully that makes sense. Like I said, the arches, they're the jack of all trades. They're kind of made up of a little bit of everything. All right. Question 6. So this is going to be more of a regional level question, I think. It was is what was said, but we're going to go ahead and ask this more clinical question. So a newborn is diagnosed with a developmental condition caused by a problem early on in embryonic development. The doctors expect that this error is going to affect structures that normally develop from the inside lining of the throat. Which structure is most likely to be affected? Okay, I'm going to go ahead and end this poll. The correct answer is D, so good job. Y'all are doing really great. So the structure that's going to be affected is going to be the pharyngeal pouch. So the reason why is because we said it's going to affect the inside lining of his throat. So we're looking at the inside structures. That's going to be the pouches. Sorry. So in summary, just to reiterate kind of what we talked about, embryology is tough in that there's a lot of moving parts, but y'all have done really amazing based on all these questions. So let's just summarize one more time kind of what's composing the pharyngeal apparatus. So once more, we're looking at a cutout section here, just a cross section here. Which is kind of almost like we're laying it on a table, looking at it from top down. We have our pharyngeal pouches, which are made of endoderm. There's 4 of them in total, and they are found in between the arches. So on the inside is our pouches, in between the arches, and they are made of endoderm. Eventually, they're going to become your glands and your inner linings, which we talked about a little bit in the prior question. So he had a developmental abnormality on the inside of his throat, so that obviously came from his pouches. Our mesoderm component is going to be the core of the pharyngeal arch, and it is going to be all 5 of them, remembering that arch 5 goes away, and so this is going to be arch 4 and arch 6. It's going to support and help comprise movement, so muscles, bones, and neural crest cells, and it's also going to become your nerves. So how I kind of like to remember, and I know that this is getting more into regional, so you don't have to worry about this right now, but a good mnemonic that I use in order to remember that our mesoderm supports like our movement and muscles is that our arches, our arch 1, which is made of mesoderm, also makes up all of the things that start with an M. So I'm just going to list off a few of them, but you don't need to know this right now or to this detail, I don't think. So your muscles of mastication, so the facial muscles that help you chew, your mastoid air cells, maxillary artery, all of these things start with M, and they come from the mesoderm. Your ectoderm is going to be the external surface, which is going to be the external pharyngeal arches and your pharyngeal clefts or grooves, same thing. And your groove 1 here is going to become that outer ear, just like we said. But as you can see here, some of those things are more regional. And so just knowing that this is your endoderm, your mesoderm, and your ectoderm is really great. Again, this is going to be happening in embryogenesis, so occurring through weeks 4 and 5, and then this more structural development is going to happen at weeks 6 and beyond. So it is important to note, just in case I didn't say it yet, your pharyngeal apparatus is going to be this whole structure, but it is bilateral. So you do have arches, clefts, and pouches on both sides. And with that, I think that is it. Those are my resources. So if anybody has any questions. Let's listen. I don't see any, so. All right. Well, I will there's a question. So could you re-explain the changes occurring to the pouches? So what changes are you specifically talking about here? Goodness. You mean like the development and why, how it's numbered? Like, oh yeah, okay. So this, without getting too into the details, just knowing, let me find a picture that labels them here. There it is. So without getting too much into the details, it's not something that has to be too overly complicated. I just wanted to point out, in case you were curious, why we jump from arch 1, 2, 3, 4, and then we jump to 6. That's just because all of these arches, they're numbered because they're going to develop into an adult structure. Arch 5, it doesn't develop into any adult structures, it just goes away. So they don't include it in the official numbering. That's just the only thing I wanted to point out there. Does that clear that up, and does that make any sense? Yeah, no problem. Okay, well, I will also drop my email in the chat. If anybody has any questions, feel free to reach out. If anybody thinks of anything, I'll also be here for a few more minutes. Thank you.
Okay, are there any further questions for our presenters this evening? Okay then, Dr. Mapes, did you want to have any closing remarks before we end this evening?
Good. I
Okay.
think I said all I needed to say at the top of the hour. Oh, looks like okay, now it's Savannah's email. Yep, we're good.
Okay. All right. Well, everyone have a lovely evening and best wishes to you. This might be the last time I see you before the competition, so I wish you all the best and congratulate you on all of your hard work and dedication, and look forward to hearing how things happen at the competition. So, best wishes to everyone. Happy New Year.